Today we'll discuss one of the important topics from biotechnology and applications, that is RNAi or RNA interference. So we'll discuss in a very simple way. So this is one of the toughest topics of biotechnology applications. So let us discuss RNA interference. So what is the need of RNA interference? It's a novel technology or a very commonly used technology to produce genetically modified plants. So the plants with RNA interference technology are this novel technology. The plants can protect themselves from different parasites or insects, so based on the specific genes. So this technology discovered by two scientists called Fire and Mello in 1998. So who discovered RNA interference technology? It is all about Fire and Mello. Fire and Mello, they discovered in 1998, and for this invention they got Nobel Prize in 2006. The plants with RNA interference, they become a pest resistant. Why? Because we are introducing a specific genes to the particular plant. So it is all about the plant can protect themselves from the parasites or insects. So it is all about a genetically modified plant, which is we are commonly say that it is all about a pest resistant plant. Now, who is pest here and who is that plant? So we are discussing about the pest. It is all about a nematode, a pathogenic nematode. It is all about a Melidogyne incognitia, which causes a root knot disease in tobacco plant. So when it comes to this pest resist plant, who is pest here? The pest, it is all about Melidogyne incognitia. The plant, it is all about tobacco plant. This Melidogyne incognitia or MI, this causes root knot disease. What is the disease name? The disease name, it is all about root knot disease in tobacco plants. So when it comes to these knots are commonly called as feeding sites. These knots are commonly called as what? These knots are commonly called as feeding sites. From these feeding sites, the Melidogyne incognitia loots everything. Everything means what? Entire nourishment from the it loots entire nourishment of a tobacco plant. So due to lack of nourishment, lack of nutrients, there is a drastical effect on the tobacco productivity. So what is the point here? Melidogyne incognitia, which causes root knot disease. In this disease, what it forms? It forms many feeding sites. From these feeding sites, within the root cells of a tobacco plant, the Melidogyne incognitia takes entire nourishment of a tobacco plant. So there is no nutrition. There is insufficient nourishment from the tobacco plant. And who is responsible for this? Knots or feeding sites. It is all about. So it is all about the toxic proteins. The toxic proteins of a nematode, which is that Melidogyne incognitia. The toxic proteins of Melidogyne incognitia that is responsible for the feeding sites. So what is the source of these toxic proteins? It is all about the mRNA of nematode that is responsible for these toxic proteins. Again, it is a very simple. We can say that the central dogma of any organism that is responsible for the protein synthesis. The Melidogyne incognitia DNA that send a message to mRNA. That is what is called transcription. From mRNA is responsible for the protein synthesis. That is what is called a translation. So here it is all about the translation process that is responsible for the toxic proteins. These toxic proteins are responsible for feeding sites, which is the major symptoms of this particular root knot disease in tobacco plants. Now, what is the point here? So, with the help of RNA interference technology, so we can silence the mRNA, or we can interfere with the mRNA translation process by degrading the mRNA of nematode. There is no mRNA, no toxic protein, no feeding sites, no effect on tobacco plant. So without feeding sites, the Malidogony incognitia can't survive in the tobacco plants. So it is all about this technology. It is all about by degrading, by damaging the mRNA of the nematode, so we can prevent the plant from the diseases. So it is all about, simply we can say that by breaking down the mRNA, by degrading the mRNA of nematode parasite, so we can prevent the plant from the this particular parasite. How we can degrade this mRNA? That is all about RNA interference. So with the help of the degradation of mRNA, the silencing of mRNA with the help of dsRNA from the tobacco plant, that will initiates, so this dsRNA that initiates RNA interference or RNAi. Then what is the source of this dsRNA? Let us discuss in detail. So it is all about pest resistant plant is what? A pest resistant plant is a genetically modified plant. Means what? So this plant's DNA, this tobacco plant's DNA already we manipulated. So what is the point here? We manipulated the tobacco plant. So that is what it is called, genetically modified plant, with the help the technology called RNA interference. So what is the point here? Into the tobacco DNA, we manipulated in the tobacco DNA. So we inserted some nematode specific genes. What is the point here? Nematode specific genes from naturally from the some plants are very much resistant about nematode parasites. So we'll take the genes from those plants and we insert into the this particular tobacco plant. It is all about nematode specific genes introduced into the tobacco DNA. Now the tobacco plant becomes a pest resistant plant. So nematode specific genes are introduced into the tobacco DNA by using agrobacterium vectors. Agrobacterium vectors is very important for NEET exam. By using agrobacterium vectors, these agrobacterium vectors, with the help of when it's come to the plant, it is all about the vector, it is all about agrobacterium. That is very important for NEET exam. So with the help of this vector, we inserted a nematode specific genes into the DNA of tobacco plant. Now the tobacco plant become a pest resistant plant. So when Malidogony incognitia that enters into the plant, this genetically modified plant that will produce this GMP plant or pest resistant plant that will produce two RNAs. One is a sense RNA, the other one is antisense RNA. One is sense RNA, the other one is antisense RNA that is produced by the nematode specific genes which are present in the genetically modified plant. So these two RNAs are single stranded. These two RNAs are what? So when a Malidogony incognitia enters into the root cells of the tobacco plant, now the plant will produce sense RNA and antisense RNA. These two are complementary to each other and forms a complementary dsRNA. This complementary DS, that is what double stranded RNA that initiates RNAi. What is the source of this dsRNA? It is all about the sense RNA and antisense RNA. They complementary to each other and they paired and forms a dsRNA. How this dsRNA that will breaks the mRNA of nematode we'll see here. So this dsRNA combined with the some proteins which are present in the cytoplasm of a tobacco root cell. So before that, within the cytoplasm of tobacco root cells, there is an enzyme called dicer. There is an enzyme called dicer, which is very commonly present in the cytoplasm of tobacco root cells. So this dicer enzyme that will break down the dsRNA and forms three different RNAs. That is what is called a small hairpin RNA and a micro RNA and a small interference RNA or siRNA. What is the point here? There is an enzyme called dicer. That will breaks the dsRNA into three small double-stranded RNA called a small hairpin RNA, micro RNA, and small interference RNA. Any one of the RNA, all the three RNAs can capable to break down the mRNA of nematode. Here, what is the point? Let us say this small hairpin RNA, this small interference RNA, siRNA means what? A small interference RNA. miRNA means what? A micro RNA. shRNA means what? A small hairpin RNA. So what is the point here? Any one of the RNA, either shRNA or micro RNA or a small interference RNA, that can combine with the certain proteins which are present in the cytoplasm of tobacco plant. They will form a complex called risk complex. What is the complex? So this any one of the RNA that combine with the proteins and forms a complex called RISC complex. That is what is called RNA induced silencing complex. What is the full form of risk? The full form of the risk, it is about RNA induced silencing complex. This RNA silence inducing complex that all about formed within the root cells of tobacco plant. What is the point here? The small interference RNA that binds with the certain proteins which are present in the tobacco root cells and forms a risk, that is all about RNA induced silencing complex. So this risk, this RNA induced silence complex that will reach the nematode body, nematode cells through ingestion. There is something called, there is a connection called a plasmodesmata. So with the help of by ingestion or plasmodesmata, what is the point here? It is all about when this Meloidogen incognitia that it lives in the root cells of the tobacco plant. When this risk complex is there in the root cells of the tobacco plant, it commonly feeds in the cytoplasmic material of the tobacco root cells. So during that process, what happens? This risk complex will reach the nematode cells of nematode body. So what happens? So it is all about what we can understand here. From the formation up to risk complex, it is all about a tobacco root cell. Up to from dsRNA to risk complex, it is all about root cells of tobacco plant. So now it is all about this risk complex will reach the nematode body. So within the nematode body, what happens? So within these proteins, there is something called, there are two enzymes are there. There is something called enzyme called or protein called ergonaut protein. This ergonaut protein, this will help the siRNA to find out this particular mRNA of nematode. What is the point here? This ergonaut protein that will guide the small interference RNA to find the mRNA of a nematode. So this ergonaut helps to find the mRNA of. At the same time, this ergonaut protein that also unwinds the small interference RNA. Now the small interference RNA becomes a single stranded RNA. It becomes what? ssRNA. This ssRNA that binds with the mRNA, and again that will form a double stranded RNA. Apart from that, there is an enzyme called slicer also present in the slicer also present within the this RISC complex. And now this slicer will break down this mRNA and the ssRNA complex. Here ssRNA, it is all about antisense RNA. This is what this antisense RNA and the mRNA complex are degraded by the enzyme called slicer. So that is how the degradation of mRNA will happens within the nematode's body. So there is no mRNA, no toxic protein, no feeding sites, no effect on the tobacco plant. Now the tobacco plants, they can protect themselves from the pathogenic nematodes. So this is how RNA interference will works. It is all about only one thing. It's all about the degradation of mRNA. Degradation of mRNA with the help of the dsRNA, that is all about double-stranded RNA, which is produced by a tobacco plant. So this is how the entire process. So that will break down the mRNA with the help of a dsRNA. So there is no toxic protein, no feeding sites. Without any feeding, without any food, the Meloidogyne incognitia can't survive within the nematode's body. So this is how the plants can protect themselves from the nematode parasites. Now one more point is very important for exam. It is all about this RNA interference is very commonly seen in the all unicellular organisms at cellular defense mechanism.